If your ring finger is clearly longer than your index finger, you are carrying one of the most measured, most argued about proportions in the entire human body. For more than a century, scientists could not agree on what it meant. Some believed it was pure randomness. Some believed it tracked hormones. Some believed it tracked something older, something written into Ancestry itself. The debate filled thousands of papers across dozens of countries, and for a long time nobody could close the case. Then, in just the last few years, large-scale DNA studies finally began closing in on an answer, tracing the trait back through ancient migrations, through population bottlenecks, into a specific corner of human prehistory. What the researchers found surprised even the teams running the work, because the strongest genetic signal did not come from where the textbooks assumed it would. Now, look at your hand. The pattern itself was noticed long before anyone had a scientific way to measure it. Victorian-era surgeons mentioned it in their private letters, describing hands where the fingers lined up evenly and hands where one clearly outreached the other. Renaissance anatomists sketched it into their studies of the human hand, treating it as just another curious variation worth recording. Even further back, in classical Greek and Roman writing, there are scattered remarks about hands that appear balanced, index and ring finger reaching nearly the same line, compared with hands where the ring finger pushes visibly past the index. Nobody at the time had any framework for explaining what they were looking at. They simply noted it, the way they noted other quirks of human appearance. And the observations sat quietly in libraries for centuries, waiting for the science to catch up. The modern study of the trait began with something deceptively simple: a name and a formula. The second digit is the index finger. The fourth digit is the ring finger. Divide the length of the index by the length of the ring, and you get a single number, the digit ratio. If the two fingers are the same length, the ratio is 1. If the index is longer, the ratio rises above 1. If the ring finger is longer, the ratio falls below 1. That third case, the ratio below 1, is the one this video is about. A small detail, measured in millimeters, that turned out to carry an enormous amount of hidden information. The modern breakthrough on this trait began with a British researcher named John Manning. In the late 1990s, Manning started publishing systematic studies of the digit ratio, measuring thousands of hands across different ages, different sexes, and eventually different populations around the world. What he found was striking. The ratio is set very early in life, almost certainly before birth, and once it is set, it stays fixed. It does not shift in childhood, it does not shift in adulthood, it does not shift in old age. Whatever shapes the ratio does its work quietly in the womb, locks the proportions of the developing fingers into place, and then leaves a permanent record on the hand for the rest of a person's life. That permanence was part of what made the trait so compelling to other researchers. Unlike muscle mass, weight, or skin tone, the ratio cannot be changed by lifestyle. It cannot be trained. It cannot be earned later through effort. Whatever the body recorded, it recorded once, before birth, and never again. The leading explanation for what shapes the ratio is prenatal hormone exposure. Between roughly the 8th and 14th weeks of gestation, while the bones of the fingers are growing rapidly, the ring finger appears to respond strongly to testosterone present in the womb, while the index finger responds more to estrogen. Higher relative testosterone tends to produce a longer ring finger, a ratio below 1. Higher relative estrogen tends to produce an index finger that matches or exceeds it. This is the chemistry the hand quietly remembers. For years, that hormone explanation was treated as the entire story. Ratio reflects the womb, case closed. But the story didn't stay closed for long, because as researchers began measuring hands outside of Europe and North America, a strange consistency appeared. Within any single population, the average ratio held steady, study after study, but between populations, the averages didn't match. Different countries, different ethnic groups, different ancestral backgrounds produced different baseline numbers. The differences were small, usually only a few hundredths of a point, but they were not random. They repeated. And one of the strongest, most consistently confirmed patterns in the entire body of research came from East Asia. Studies carried out in Beijing, Shanghai, Hong Kong, and Taipei, in Tokyo, Osaka, and Kyoto, in Seoul and Busan, and across dozens of smaller cities and rural districts, kept turning up the same finding. On average, individuals of East Asian descent showed a digit ratio slightly lower than the global average. The ring finger reached a little further past the index than it did in most other populations measured. It wasn't a difference you could spot by glancing at a hand. But across large samples, the East Asian average consistently sat lower than the European average, lower than the African average, lower than several other regional averages in independent studies done by independent teams across multiple decades. For nearly 20 years, that finding sat in the literature as an unexplained footnote. Nobody could say for certain whether it was genetic, environmental, or simply a quirk of measurement. The question hung open for almost 2 decades, until a wave of large genetic studies, conducted between the late 2010s and the early 2020s, finally began supplying real answers. The breakthrough came from genome-wide association studies, known as GWAS, which scanned the DNA of tens of thousands of people at once, searching for genetic variants that correlate with a measurable trait. Researchers built GWAS studies specifically around the digit ratio. They measured enormous cohorts of hands, pulled the DNA behind them, and ran the comparisons. What they found was that the ratio is highly polygenic, shaped not by one single genetic switch, but by many small variants working together. Still, within that tangled polygenic signal, certain genes kept surfacing as central players. At the top of the list sat the HOX genes, specifically HOXA13 and HOXD13. These are ancient genes, deeply conserved across vertebrates, and their job is to shape the bones of the digits during embryonic development. HOX genes are among the oldest working genes in the human body, already active in the ancestors of fish hundreds of millions of years ago. In humans, they orchestrate the basic layout of the limbs, the patterning of fingers and toes, and the precise timing of bone growth in the developing embryo. A slightly stronger pulse of HOXD13 in the ring finger region, a slightly weaker pulse of HOXA13 in the index finger region, and the hand that emerges has a ring finger reaching noticeably past the index. Shift that balance the other way, and the fingers line up evenly instead. The genetic variants that nudge this balance are not rare. They are scattered throughout human populations everywhere on Earth. But the frequency at which different variants appear is not the same from one part of the world to another. And that is where the question of ancestry comes into sharp focus. When researchers examined the population frequencies of the variants most strongly linked to a lower digit ratio, they found that several of these variants are notably more common in East Asian populations than in European or African populations. Not exclusive to East Asia, not found only there. But the percentages tilt in a consistent direction. A variant that appears in roughly 15% of one population might appear in over 30% of another. Another variant, present at modest levels almost everywhere in the world, sits at noticeably higher frequency across Han, Chinese, Japanese, Korean, and related groups. These are the numbers behind the pattern. The population frequency differences in these specific variants are what produce the average ratio differences researchers had been measuring in hands for decades. The hand was never lying. It was simply reporting an underlying genetic distribution that had been quietly present in the population the entire time, long before anyone thought to measure a single finger. The next question was the deeper one, and it was the one that would take researchers out of modern genetics entirely and into the deep past. Where did these variants actually come from, and how long had they been sitting in these populations? To answer that deeper question, scientists turned to ancient DNA, the field that has quietly revolutionized the study of human ancestry over the past 15 years. By sequencing genomes recovered from skeletal remains, some of them tens of thousands of years old, researchers can compare modern populations directly against the ancient people who actually lived in a given region. They can ask which variants were already present in which ancestral groups, and trace a single trait backward through prehistory with a level of precision earlier generations of anthropologists could never have imagined. What the ancient DNA evidence suggests, once combined with the modern population studies, is that several of the key digit ratio variants are genuinely old. They were present in the populations that moved out of Africa more than 50,000 years ago. They were present in the early modern humans who pushed across Central Asia and reached the eastern edge of the continent by roughly 40,000 years ago. Some of these variants even appear in the genome of the Tianyuan individual, a man whose remains were recovered from a cave near Beijing, who lived around 40,000 years ago, making him one of the earliest modern humans known from East Asia. The variants did not appear after his time. They were already there when his population arrived. They came in with the first wave of modern humans to reach the region, and they simply stayed, passed forward, generation after generation, into the populations living there today. The picture that emerges from all of this combined evidence is one of slow, patient accumulation rather than any single dramatic event. As different populations moved into different regions of the world over tens of thousands of years, the frequencies of these variants gradually shifted. In East Asian populations specifically, certain variants linked to a lower digit ratio rose to higher frequency over time. Why exactly this happened is still genuinely debated among researchers. Some point to genetic drift, the random fluctuation of allele frequencies that occurs naturally in any population, especially during periods of small population size or during expansion into new, unfamiliar territory. Others point to subtle selection pressures, possibly tied to climate, diet, or specific conditions affecting fetal development in certain environments. Others still suspect a combination of both, drift setting the initial stage, weak selection nudging the frequencies further across many, many generations. Whatever the exact mechanism, the result is what shows up in the measurements we take today. If you carry the trait of a clearly longer ring finger, the genetic ancestry underlying that trait points, in many cases, toward a deep East Asian lineage running somewhere through your family tree. That does not mean every person with a lower ratio is of East Asian descent, because the variants exist in every population on Earth. What differs is not presence, but frequency. And frequency tells its own quiet story about where a trait was preserved most strongly across the long history of the species. East Asia is one of the strongest reservoirs of this trait, but it is far from the only one. The indigenous populations of the Pacific form another, with Polynesian, Micronesian, and Melanesian groups showing patterns consistent with East and Southeast Asian ancestral input reaching far out across the ocean. Parts of South Asia, shaped by ancient migrations that shared deep genetic heritage with East Asia, form another reservoir still. Southeast Asia carries a particularly layered version of this story. Long before more recent migrations arrived, an earlier wave of modern humans, sometimes called the Hoabinian foragers, reached the region tens of thousands of years ago and left their genetic mark. Much later, a second wave arrived, associated with the spread of rice agriculture and the Austronesian language family, moving out of southern China, through Taiwan, and outward across the islands of Southeast Asia and into the Pacific. Both waves carried digit ratio variants with them. Both waves contributed to the gene pools of the populations living across the region today. When researchers examine the genetic ancestry of someone from the Philippines, from Indonesia, from Vietnam, or from Malaysia, they typically find contributions from multiple ancient populations layered on top of each other, and the digit ratio variants travel along with that ancestry. The hand records the layering, even though it cannot tell you, on its own, which wave contributed which fraction of the final proportion. For viewers of Korean and Japanese descent, the picture carries its own distinct layers. Modern Korean populations show strong genetic continuity with the ancient populations of the Korean peninsula, layered with additional contributions from neighboring regions accumulated over thousands of years. Modern Japanese populations carry an even more complex mix, including the older Jomon population, which lived across the Japanese archipelago for tens of thousands of years, and the later Yayoi wave, which arrived from the Asian mainland only a few thousand years ago. Both the Jomon and the Yayoi populations carry digit ratio variants. The Ainu of northern Japan, who carry stronger genetic continuity with the ancient Jomon line than the rest of the modern Japanese population, have been studied separately in smaller digit ratio surveys, and the results align cleanly with the broader East Asian pattern already established. The modern Japanese genome reflects the blending of these two ancestral streams, and the average digit ratio measured in the population today reflects their combined contribution. For viewers of Chinese descent, the genetic history reaches deeper still. Northern and southern Chinese populations show genuinely distinct genetic patterns, shaped by different ancient ancestries and different migration histories across thousands of years. Both, however, share many of the digit ratio variants in question, and both feed into the broader East Asian average that researchers have been carefully measuring for decades. The Han Chinese population, given its size and its long history of expansion, has been one of the most heavily studied groups in this field, and its consistently lower ratio has been one of the strongest signals driving the patterns seen worldwide. Before I show you the last part, if you want to look at your own results and see where East Asian ancestry fits into your bloodline, I put a full 3-volume PDF bundle over at the Origin DNA website. It shows you how to read your raw data and find matches beyond what Ancestry or 23andMe tell you. Link is in the description. But stay right here, because what comes next is the part most people completely miss. It is worth pausing here to address what this trait does not mean, because popular writing has badly overreached on this subject for the better part of 20 years. A lower digit ratio is not a measure of intelligence. It is not a reliable measure of athletic ability, despite popular claims to the contrary. It is not a measure of personality, sexual orientation, financial success, or romantic prospects, despite a long history of weak, poorly controlled studies that tried to link it to all of these things. The serious genetic literature is clear on this point. The ratio is a developmental marker, shaped by prenatal hormones and by a polygenic genetic signal inherited from your ancestors. It tells you something about the chemistry of the womb you developed in, and something about the variants passed down through your family line. It does not tell you anything about who you are as a person, what you are capable of, or what your future holds. Reading too much into the trait, in either direction, misses the actual point of what the careful science has found. Look at your hand again. The ring finger that reaches past the index, the proportions locked into place long before you had any memory of your own body. The variants present in populations that walked across continents thousands of generations before you were born. None of it was chosen, none of it was earned. It was simply given, quietly, in the womb, and it has stayed on your hand ever since. The trait is not loud. It does not announce itself. Most people go a lifetime without ever thinking about the relative length of their fingers. But now you know what the proportion is actually recording. A fragment of fetal development, a fragment of inherited genetics, and in many cases, a fragment of an ancient East Asian lineage that walked out of Africa, across Central Asia, and into the river valleys and mountain corridors that became the region we know today. Your hand is older than any of that history. It is one of the few visible records the body carries of a story otherwise hidden entirely. That's how this lineage spread. But if you want to go beyond basic percentage charts, go to the Origin DNA linked below. You get a full 3-volume PDF bundle that teaches you how to read your raw data, use matches to find relatives, and uncover the deep history Ancestry and 23andMe leave out. Click the link in the description and unlock your full results right now.